we thought that saying just yes or no qualitative may not be enough for this disease and other diseases that we deal with. So we also impose a quantitation of the viral load, which we can talk later on, but we think that it has been important. And in then the context of following and monitoring the patients, not for diagnosis, we have the test that we are discussing today, which is a test that is based on the detection of the antibodies that we produce against this foreign virus, this foreign aggressor. Nature has given us an attacker, but we also have, a good development in producing the attackers, kill, and it's the antibodies. These antibodies can be quantified, also not just being in a qualitative assay. So the difference between our assay and assays that are in the market is that it's an assay that is based on chemistry. It identifies antibodies in the serum. For the reason this technology is called serology. And we can do the same that we did with the viruses. We can qualitatively say yes or no, but the difference between the assay that we have and other assays that are in the market is that the assay of Dr. Kramer allows us to produce two assays in one. The first is to say yes or no, qualitative, but the next one allows us to do a quantitation, a titer, to know how rich your blood is in the context of how many antibodies you may have. And that has been a differential from our test compared to the other tests. Based on the fact that it's two tests in one, it's highly specific and highly sensitive. And these are important connotations. Sensitivity deals with the false negatives, but in here what is important is the false positive, the specificity, because if I say that to somebody that it's positive when in reality it's negative, and we allow this individual to go into the front line, for example, chances to be sick are very high. So our test is 100% specific and highly sensitive.